Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really awesome guest today involved in science, technology that's creating a better tomorrow. Today on the show, we have the honor of being joined by Josh Haimson. Josh is the co-founder and chief executive officer of a company called Inductive Bio. They are a tech company. They're focused on democratizing artificial intelligence models to ultimately transform small molecule drug discovery, focused on eliminating so-called ADMET or absorption, distribution, metabolism, excretion, and toxicology bottlenecks with their state-of-the-art AI models and generative chemistry, ultimately powered by this really interesting pre-competitive data consortium model, which we'll be getting into. Josh has spent his career focused at this really interesting intersection of machine learning, products, as well as life sciences and healthcare. Prior to Inductive, Josh was the director of product for the machine learning and data curation organizations at Flatiron Health. There, his team worked to generate real-world evidence at scale across their network of several million active cancer patients for use by researchers in pharma, academia, as well as government. And prior to Flatiron, Josh was at MIT studying computer science, working with researchers at Mass Gen Hospital using machine learning and natural language processing to predict things like patient response to cardiac resync therapies, as an example. A lot of really cool themes to get into today, and honored to have him with us. Josh Haimson, thank you so much for taking time to come on the show.
Thanks for having me here. It's really great to get to have this important conversation.
Absolutely, absolutely. I look forward to it. Just let's start off back at MIT because I took a look back, as I mentioned. You were involved in cardiac resynchronization therapy and sort of using machine learning at the time to not just think about what types of patients this may be good for, but the ones that it's not good for, who may benefit, who may not. Take us back a little bit to sort of the early days, CSAIL, and a little bit of what got you initially interested and focused on this really relevant area in 2025.
Yeah, definitely. In some ways, it really kind of starts a couple decades before that, actually. It really, when I was just a few days old, my parents had just brought me home from the hospital, and during my first pediatric checkup, doctor noticed that my resting heart rate was in the 200s. And so I was rushed back to the hospital, went through this battery of tests, ultimately got diagnosed with supraventricular tachycardia. And to stabilize my heart rhythm was prescribed a cardiac glycoside drug called digoxin. And so for the first year of my life, my mom gave me this really precisely measured dose of digoxin, which, as I later learned and sort of implied by the last half of that word, is a really narrow therapeutic index and can be really toxic if it's given at slightly higher doses. And thankfully, my heart stabilized. I was able to come off of that drug. And fast forward a couple decades, I was doing AI research at MIT, increasingly became fascinated more intellectually by how all these advances I was seeing in machine learning to identify patterns in massive amounts of data and make sense of it could really intersect with the massive amounts of data that were coming out of the omics revolution in biology. But I got really inspired to focus much more pragmatically on translating that into, real world impact on patients, and kind of started by digging into my own medical history and digging into digoxin. Learned that the use of that drug dates back to at least the 18th century. An English physician named William Withering published one of the first really empirical studies of the safety and efficacy of that drug in hundreds of patients, and ultimately just blew me away that who had died centuries before I was born could have such a really profound impact on my health. And that really kind of inspired me, to have this lifelong goal of leveraging the tools that I was good at in developing, which is around AI and machine learning, to really treat disease for many generations to come.
Mhm.
So that's kind of what pushed me down this path. And at MIT, it was a really interesting set of work where we were working in CSAIL. I was working sort of in two labs. One was a much more theoretical AI lab in Patrick Winston's lab, and then a much more kind of applied machine learning work with Regina Barzilay and Mass General. Um, and it was fun to see that intersection sort of carry through to now and all the advances over the last decade, where those are really starting to come together and make meaningful impacts in patients' lives at the end of the day. So it was a really good formative experience of being able to take these tools that have been advancing at a really rapid rate and applying them to really important problems for patients.
Outstanding, outstanding. Yeah, it's funny. Just as a side note, we could talk offline about this, but it's very
Mhm.
interesting. You wonder if you fed digoxin, which is a cardiotoxin. I mean, it's what it is. Discovered by the old school natural product route. You wonder if you fed it into one of these high tech AI models that's related to toxicology, whether it gets thrown out early or what happens to it. But I guess that's for another show and another discussion. But yeah, I mean, it's really interesting that looking again at some of your work at Flatiron, and there was an interesting piece I pulled up. It was a presentation that you actually gave back in 2018 at the Flatiron Research Summit. At the time, you're talking about this evolving role of machine learning. You went through some of the sort of the core principles. We had Blythe Adamson on several months ago talking about sort of their work in cancer. And in this presentation, you go into this theme of so-called technology-enabled abstraction, the sort of this issue that, hey, this technology is awesome at what it does, but we need to pair it. We need to pair it with important people, depending on the discipline. In the specific case, we're talking about clinical abstractors. We'll be getting into, when we get to inductive and chemist in a loop and all that stuff, talk about that importance. But talk about some of your learnings at this point, because, the intersection of this important technology enabled abstraction with the importance of the large training data sets, which you also talk about during this presentation, very integral to sort of how this all fits together. I was wondering if you could take us back a bit, talk about some of the key learnings from that period.
Yeah, absolutely. I think ultimately it's a theme that has really carried through my entire career, which is being incredibly pragmatic about what these tools can and can't do, and then building systems around them to get a lot of the benefits from what they can do while kind of mitigating the downsides of what they can't. And at Flatiron, this was it feels like ancient history in AI timelines now. But the state of the art in natural language processing that we were working with at the time was really good at getting to the 95, 96, 97% accuracies, but getting that last, 3 to 5% is really, really hard and still is today to some extent, depending on the domain, even with the advances in large language models. And so what we spent a lot of time at Flatiron thinking about is how do you leverage the technology both in its current state and in the state where it's headed to get a lot of value in terms of being able to extract much more amounts of information from these medical records to do really important research, but kind of mitigate that risk, knowing that some percentage of the time it's going to get it wrong. And we have a similar kind of challenge in medicinal chemistry where these AI algorithms can design really interesting molecules, but they're not always good molecules. And so that's really where I focused a lot on pairing real world human expertise with these tools and really treating them as tools that can supercharge those experts rather than tools that are designed to, ultimately try and replace them.
Mm-hmm.
There's a classic quote, I think it's usually attributed to George Box. I'm not sure if he actually said it, which is that all models are wrong, some are useful. And I think that really kind of guides our philosophy and how we build these tools, which is that at the end of the day, they're just models and they're going to be wrong some percentage of the time, but they can do lots of things that humans can't. So if you pair them with experts and do it thoughtfully, you can get the best of both worlds that you really can't get either with either alone.
Mm-hmm. Excellent, excellent. So yeah, let's talk a little bit about inductive bio, and we'll be getting into your really unique models in a teeny bit. But I'd love to just start off, as I mentioned in the bio, the core focus of what you're looking at there is, sort of this intersection. We used to call ADME, now it's called ADMET, with the toxicology put on there. So sort of everything pharmacokinetics plus toxicology merged in there. We don't normally think about it. We know, okay, this is tremendous percentage of drugs that just fail, that enter clinical trials that fail, something like 90 some odd percent. We don't maybe think as much about the fact that a lot of that failure is just attributed to the fact that, we get our kinetics and toxicology wrong early on. Some studies show them even a much higher rate than that. Talk a little bit about ADMET. And just, for our audience that may be less familiar with it, why this is such a crucial step in the drug development process in general. And then we'll go into what you're doing about it at Induct.
Yeah, absolutely. I think, if you think about what a drug has to do, it actually goes on this really crazy journey through the body. And it's in some ways it's just amazing to me that we actually can design drugs to have the effect that we want. If you think about it, when you take a pill, it has to survive the acids in your stomach, it needs to make it into the intestine, it needs to be permeable enough to cross the lipid membranes and get through the intestinal wall into the bloodstream. It needs to be soluble enough to even dissolve in that bloodstream and start circulating throughout the body. Your liver has a whole bunch of enzymes designed to try and remove anything foreign from the body to protect you. And so it sort of needs to dodge that detox system in your liver. It needs to, if you're targeting something in the CNS, it needs to cross the blood-brain barrier. If it's an intracellular protein, it needs to cross the cell membranes. It then needs to reach that right protein in the right cell at the right tissue at a high enough concentration to have the effect that you want it. And it needs to do all of that while avoiding hitting every other protein in your body that if it, bound to could cause some toxicity issues. And so this wild journey that pill takes on, it's a little bit reduction, but absorption, distribution, metabolism, excretion, toxicity, that acronym of ADMET really kind of encompasses everything that the drug needs to do in your body, with the exception of hitting that one protein target. And I think we ended up focusing the company on this really after a long research journey, talking to hundreds of researchers, really with kind of one question in mind, which is there's all this hype around AI and drug discovery right now. Where can the state-of-the-art machine learning actually move the needle? And we talked to hundreds of researchers, ended up spending a lot of time with medicinal chemists who are the scientists that are designing these new drug candidates. And they kept using the same phrase to describe their work, which was this phrase, whack-a-mole. And when we dug in and understood, what is it that you're doing? What are the challenges you're running into? It ended up being playing this game of whack-a-mole between all of these different ADMET properties, where over the course of a typical drug program, you'll make thousands of molecules over years to identify one that has that right balance of potency and ADMET to actually have a good shot of working in humans. And that really has a critical impact in, as you said, the likelihood of success of that drug at the end of the day. If you don't get the pharmacokinetics of that drug profile right, you might not get the level of exposure that you want in, a brain tumor, for example, to actually be able to kill those cells. If you don't get the toxicity right, you'll obviously cause safety issues with a drug. And so we just saw it as this massive problem, but it was also one where we were starting to get a lot more data because unlike targeting a specific protein, the human system is the same across any drug program, whether you're targeting oncology, whether you're targeting CNS, inflammation. Ultimately, you still have to deal with the same enzymes in the liver. You have to permeate the same intestinal wall. And so you could start to build up these large data sets, which are really key for building effective machine learning models. And we were starting to see kind of the early signs of this working when we were at Flatiron. We had just been acquired by Roche and worked closely with Roche and Genentech, and really saw a lot of these early generations of machine learning starting to leverage the data that they had internally to help increase the odds of success of the molecules that their chemists were making. And then when we kind of went to the broader biopharma industry, we saw this really big opportunity to build kind of the next generation version of that platform, but do it for the industry as a whole rather than within one specific pharma company. So that ultimately led to Inductive, and we focused a lot on this ADMET challenge and really the data behind it, because the data is key for building anything useful in machine learning.
Hmm. And so, continuing along, the thesis. So, obviously, you pointed out we need the large training data sets, but wait a second, this AdMet data is distributed across every pharma company out there, thousands of biotech companies, every university lab that's ever worked in this area. So there's a lot of great, diverse, high quality data out there. We got to bring it all together so it benefits all of us at this important step so we can get rid of this 90% failure rate. Talk a little bit about this pre-competitive consortium model. I think it's fascinating. And then I guess you can work Beacon One into it, which is basically how you then take that consortium and the data and start feeding it into your AI systems.
Yeah, absolutely. So, we went through this journey, we honed in on this problem of AdMet, and then the question is, how do we start building models that can actually help chemists understand what the likely AdMet properties of molecules are before they spend a month in the lab synthesizing a molecule and running the experiment? And the problem you immediately run into is this data is not out there in the public domain. There's some of it out there. There's even less of it out there that is kind of really high quality and consistent and useful for machine learning. And most of it is really sitting within many different pharma companies, kind of siloed within their four walls. And so what we did was really set out to say, is there some both technological structure and also legal structure that would allow these companies to be able to share that data in the right way, such that the IP of those molecules is completely protected. That's, absolutely essential. If that IP is not protected, none of this works. Um, the confidentiality is protected. No one can see, what chemical space went into the models, but we can train these kind of large foundation models on top of that much richer, much larger, much more diverse set of chemical space that sits across all of these different companies rather than just what's out there in the public domain. And it seemed crazy at first, and a lot of people told us, pharma would never share. That's just not how the industry works. But I think the more we dug into the specifics, especially with our early pharma partners, um, the more I think we all realize that collectively this is a pre-competitive problem. It's not the type of thing that if your competitor is able to design a molecule that is metabolized slightly differently from you, that's not going to kind of lose, you're not going to lose the race to them. But everyone is spending years and years of their preclinical drug discovery timelines trying to fix all of these properties. And so there's an opportunity here for kind of a rising tide lifting all boats type of approach. And so we got kind of a few early partners to kind of all buy in at the same time to say, hey, we have a lot of this historical data. Let's contribute it. Let's use that to kind of train a model in exchange for getting access to that model that has seen a much broader and much more diverse set of chemical space. And that really kind of got this consortium model rolling where our partners can now access models that are much more performant than anything they could build internally because it's been trained on this large consortium data set.
And Beacon One, is that the system? I mean, how does it, if I'm just Pfizer and I'm coming to you today to work with you, what is Beacon One? How do I access it? How does that all work?
Yeah, great question. So the when I think about machine learning, there's sort of the foundation of it, which is the data, which we talked about, this pre-competitive AdMet data consortium.
Mhm.
Machine learning is a garbage in, garbage out problem. If you don't have good data, you can't build good models. So we just talked about the consortium approach to both to build that data set. One other thing I'll call out there is that that's not the only data that we use. So we also generate data to fill in gaps in that data set because no data set will cover all of chemical space, even across all of these different pharma companies. So we also do run wet lab experiments to kind of fill in the gaps there and continually create better and better high quality data sets. So that's the data layer. Next, we have models that are trained on top of that data to look at the structure of a molecule and be able to predict, what is the most likely pharmacokinetic profile of this molecule? How rapidly will it be metabolized? Will it be able to permeate these intestinal membranes and absorb into the body? Will it be soluble enough to actually work? All these really kind of core ADMET properties that are essential for drug design. Their Beacon is this AI model architecture that we've developed to really learn from all of this very rich, but also very heterogeneous ADME data that gets generated in the routine course of drug discovery, and be able to predict that with state-of-the-art accuracy. So Beacon is the model architecture. We're on the first generation of that. We're actively working on Beacon 2 now. But we've seen with this first version of the model and that combination of that data set, the model is performing kind of at the state of the art of the field. There was a recent blinded competition called Polaris to really benchmark ADME models and kind of the current state of the field. And our Beacon 1 model came in first place out of the 39 submissions to that blinded all-comers competition. So that kind of validates that by bringing together a lot of this really high quality but really diverse data, and then building custom-built model architectures to the problems that they're trying to predict around metabolism and permeability, you can really get kind of state-of-the-art results. So that's the Beacon model. Um, the next layer up then is uh, we think of it as the last mile problem. We saw this a lot early on as we were doing research for inductive. There were some companies that had really powerful models, but when you talk to the scientists who were actually designing molecules, they didn't even know that those models existed. There's sort of this last mile problem of, we built a great model, we validated it, we have some interesting metrics on it, but in practice, if it's hard to use and integrate into the day-to-day drug discovery process of a very busy scientist who's looking at a lot of data and making a lot of decisions, it just doesn't have any impact whatsoever. So that ultimately led us to develop Compass, which is kind of the next layer of that hierarchy on top of it, which is a software tool to help chemists use AI models, including our Beacon models, to actually design molecules and get real-time feedback from them and make good decisions with it.
And this is so this feeds into the sort of the as we were talking before about this principle of technology enable abstraction, sort of this chemist in the loop component, and again, it's quite impressive here. You talk about how during a typical design, drug cycle chemist may see like 100 different ideas, but the platform now allows 1,000 times more in these cycles, really showing the scalability and the benefit of having these large data sets. Could you just say a few more words about chemists in the loop? I think this is
Yeah,
really
absolutely.
important.
So this is then kind of the next tier up of saying you've got data, you've got models, you've got software for chemists to use those models. Chemists in the loop is then how we actually bring our own medicinal chemists to the table to help our partners actually use those models to their fullest capabilities. And so here we're really working hand in hand with our partners on their drug programs. And we're bringing designs into each design meeting that are the result of not, looking at 100 ideas, which is what a human chemist might consider in a given week, but looking at millions of ideas. So as a concrete example, we just ran this last week. We have a partner that we're working with. They have kind of this classic problem of a molecule that is potent against their target, but it is really rapidly metabolized by all these enzymes in your liver. And so, in theory, if this molecule could reach the target, it would have the therapeutic effect that they want. But the liver is going to chew it up so quickly that it's never actually going to survive in the body long enough to actually work as a drug. And so, what we're able to do with this chemist in the loop approach is say, okay, we know that this set of molecules over here are potent against this target. Let's bring both our med chem intuition. We have a chemist on the team who spent 30 years at Lilly. He's a really veteran drug hunter, on kind of what regions of the molecule are likely driving this, what could be a potential path forward here. But then they work with an AI engineer and our generative AI platform to go design, in this case, it was about 13.5 million different hypotheses for different tweaks to that molecule that we might be able to make that are predicted to kind of move the various properties forward and balance these. And then through that, we then were able to use the models to kind of whittle that down from 13.5 million, which no human could ever possibly look at, to a reasonable set of hypotheses, in this case, a few hundred compounds that all are diverse from each other and look like they are predicted to kind of improve a lot of these different properties in interesting ways. And then we bring back that human to say, okay, these are based on my experience as a veteran chemist, these are the molecules that look really, really interesting. They have specific kind of design rationales that make a lot of sense to me as a chemist, and we should kind of move those forward to actually make and test in the lab.
Mhm.
So that's kind of that chemist in the loop approach where you really, again, get that best of both worlds, where you have that deep intuition and experience guiding the program and the designs. You have the generative chemistry that can go look at millions more ideas than a human possibly can because they have to eat and sleep and spend time with their family. Um, and then you bring back that experience at the end to say, okay, these specific ideas really make a lot of sense and have a high likelihood of moving the program forward.
Yeah, one of the publicly acknowledged collaborations of yours is with a company called Nested Therapeutics.
Mhm.
And I know there was a recent paper, and we'll getting into that as sort of some of the learnings from that collaboration. I was wondering if you could just, because it's interesting, and if you can't, there's things you can't. I'm just working off the press release, so
Sure.
there's things you can't talk about. Say I can't want to talk about those. But the interesting thing about this, and just looking at Nested and sort of what they're working on here, really three things that pop right out. Number one, the difficult to treat cancer space. So the really tough ones, so melanoma and various CNS cancers and so forth. Number two, some very undruggable targets, and three, the fact that they're working on sort of this induced proximity space. So this thing where you're not really hitting a target, but you're moving things around the cell to make the existing molecules work better, and these molecular glues and all that stuff. Can you just say a couple? I think this is very, because it's really a triple whammy of really tough drug development. If you can just say a couple of words about that, any interesting learnings when you're not dealing with a compound that hits a receptor, but does other interesting types of things that we really don't yet develop drugs in this way. We're starting to, but any top line insights on that program that you could give us that's not confidential, and then we'll get into some of the learnings per the recent paper.
Yeah, it's a great question. There are a lot of details that I of course, can't go into in terms of
Understood.
mechanism. Um, I think one of the things we are seeing is these molecular glue type of programs definitely have a lot more nuance in how do you understand the activity. It's not just a competitive inhibition type of approach, and you're really trying to induce these ternary complexes that lead to degradation of your target protein um, in ways that are not always easy to predict up front. Um, but I think the field is getting there, and I think a lot of the traditional intuition and tools that we've developed to characterize molecules, to understand what is likely to work, what is not likely to work. It was interesting to see that really carry through here, and um, the the Nested team have built this really incredible platform to be able to computationally assess those types of questions. What was really exciting to see was then pairing that platform to kind of understand this really hard problem of potency in kind of this molecular glue dynamic with the ABNI platform, which then was able to bring in the other half of the puzzle that you need for that drug to actually work.
Yeah.
And kind of the combination of those two really allowed us to develop molecules that have really uh, you know, high quality compounds that have great kind of projected human dose and really balance all these properties effectively. And we're able to do that kind of much faster than kind of the typically industry standard timelines by being able to do a lot of this iteration in silico before you invest the resources in the lab to make a molecule.
Mm-hmm. It's again, it's a difficult target, difficult
Mhm.
cancers, and difficult drugs. So no, it's very impressive. Um, so yeah, I mean, so a lot of that work, or sort of some of the learnings from it, is in the ACS MedChem letters, July 25th, 2024, in an interesting paper entitled "Machine Learning ADME Models in Practice," and you talk about the need for trust building, the need for retraining. the ability, the need for these models to be interactive. An interesting point about training models on both global data, so sort of the publicly available, as well as the local, project specific data, providing the best results when you're doing this. Again, take us through. I mean, this is from several months ago, but take us through some of the sort of again the key learnings from this, and other any other learnings that in the ensuing months have popped up related to this program.
Yeah, there's definitely a few kind of really key points that we published on in that paper, but we do every day with our partners. I think one of the first ones is really rigorously assessing and pressure testing how these models are performing in the real world. One challenge as a field that kind of machine learning, especially machine learning and chemistry, has had is that there are not a lot of these realistic data sets out there to even know whether or not your model is working. And a challenge a lot of folks run into is they do what's called random splitting of molecules to test the performance of their model. The idea being, let's say we made 150 molecules and we measured all of them for their solubility. Let me train a model on a random set of 100 of those molecules and test whether or not it can accurately predict the solubility of the other 50 molecules. Um, it sounds reasonable in theory, but the challenge is that in medicinal chemistry, you're always building on existing learnings. And so if you just randomly split data, you've actually trained that model on the solution to the problem. And so it sort of memorizes the idea that, if you just add this functional group here, it will make a soluble molecule. And then when you actually do that testing process, it's able to memorize that kind of answer to the question, so to speak, but it can't actually solve that question for a new one that's going to come up. And so the better way to do it, and what we always do with our partners, is doing these time-based splits where you're trying to predict forward in time to say, let's take the first 100 molecules that we made in this program and then predict for the next 50 molecules that were made, which of those are soluble or permeable or stable. And that gives you a very different picture, actually, of which models work well and which models don't work well. We actually put out an interesting blog early on showing that if you just look at random splits, you can get kind of the completely incorrect take on which models work best and which models don't work best. So one key piece, one learning that we kind of put out in this paper was that you always really need to be doing these rigorous prospective time-based assessments of these models and then using that to inform how those models get put into practice. Because again, all models are wrong, some are useful. None of these models are going to be perfect 100% of the time. And so you have to understand where they are imperfect in order to understand how you should be using them. So that evaluation framework is kind of piece one that we spend a lot of time on, and it's really important to making this work in practice. You called out kind of this global plus local data question.
Right.
For a long time in the field of cheminformatics, there's this debate of, are global models better, are local models better? Kind of our take is use both. The current state of machine learning allows you to have models that can learn from very large, very diverse sets of data. And what we saw in this kind of second guideline that we put out in the paper was that this combination of global and local data, again, gets you sort of the best of both worlds, where a local model can kind of learn the individual nuances of a given series and a given drug program. Every chemist you talk to who's worked on a program will always kind of tell you that there's a lot of general learnings that you can get from looking at lots and lots of drug programs, but then every series has its quirks. And so that local data allows a model to learn what are the specific quirks to this individual chemotype that I'm working on. But that global data set allows you to learn generally what tends to work best across the board, and what are some of the kind of the underlying physics that tends to drive solubility or metabolism or permeability that can, you can train it in a molecular glue, but then you can go apply it to
Right.
a covalent drug. And so that combination of global and local data really allowed us to get kind of the best of both worlds, where there were some models where the model really needed to lean on that global data in order to actually make an accurate prediction because the molecule had just never been seen before in that local data. And so it had to generalize to that new area of chemical space.
Hmm.
Uh, and then there were other areas where the local data was really needed because there was a quirk of that series, and it didn't really behave the way every other drug program that kind of came before behaves with a given assay. Um, so that combination of global and local was really key. And then I think the last points are pretty consistent with the story we've discussed so far, is that it's not enough to just build good models and evaluate them and convince yourself that they're useful, but then you really need to go integrate them into the day-to-day drug discovery process. And so that means updating those models every week as new data comes in. You're always learning new things about the chemical space, and your model should be learning that as well. And then building tools to really integrate it in an interactive way with the chemistry design workflows.
Got it. No, it's extremely it's extremely impressive technology set that you put together there, and the types of things that you're exploring with it. And clearly, I mean, you just raised $25 million. I think that was announced like a couple of weeks ago. Top tier investors in both science, tech, biotech. What else has happened? What else? I know you're going to be out there at several of the ACS conferences. What else is coming up that we should know about as you get this technology package further out there, further integrated into the biotech scene?
Yeah, I think as we think about our business, I talked about the different layers, right? There's the data, then there's these beacon models on top of that. There's the Compass software that allows chemists to use those models. And then most recently, we've been developing this chemist in the loop approach where we bring, veteran medicinal chemists on our team, pair them with generative AI platforms to really kind of work hand in hand with partners on drug programs. And that is definitely something we've been developing in R&D over the past year or so. We've been seeing really exciting results that I'm hoping I can share a little bit more about soon. But really, the ability for that combination of human expertise, generative chemistry, and models that are trained on this large set of data really all coming together to move programs forward and design really high quality drugs at a rapid rate. So I'm hoping we'll be able to announce some of the results from that soon. We're also kind of scaling that up, and so you'll see a lot more on our side about how that kind of growing chemist in the loop collaboration is going to continue to kind of help support the industry.
Awesome. Awesome. Yeah, no, I look forward to continuing to follow this work. Hopefully we'll do a follow-up episode as it continues to move forward. But it's very impressive. Again, I guess again the whole ADME or the ADMET bottleneck, which I've always said is underappreciated. I'm really glad to see that you've chosen to focus here, and just very impressive work. And I wish you and your team the best with this as you continue to grow the company. But very impressive. Again, for everybody out there that's going to be listening to this episode of the show across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Josh Haimson, co-founder and chief executive officer, Inductive Bio, doing really amazing things to transform small molecule drug discovery with their really innovative AI models. Josh, again, I want to thank you for taking the time out of your schedule to talk to us for a little while today about what you're up to. Thank you for everything you do to move this area forward. As we like to say on our show, thanks for helping to create a better tomorrow for millions of patients out there via the development of these technologies. Very impressive story.
Thanks for having me, and thanks for hosting the show. It's great.
Really great having you.